Good morning, students. Today our topic of discussion is cerebellum and its internal and external neural connection. Cerebellum is formed from the metencephalon part of the hindbrain of rhombencephalon of the developing neural tube. Cerebellum or the little brain has many functions. Along with basal ganglia, it is the controller of motor system. Motor system has the hierarchical organization from motor cortex to brainstem to spinal cord. The basal ganglia and cerebellum modify motor commands from moment to moment basis. On their way to sensory cortex, the sensory pathway gives the collateral to cerebellum, so the actual sensory information are acknowledged by the cerebellum. Likewise, descending motor pathway starting from the motor cortex gives rise collateral to the cerebellum. So action plan made by the cerebrum is known to the cerebellum. It compares both the information and through its internal complex neural circuitry, it modifies and adjusts the motor activity. For proper motor activity, proper muscle tone and body balance are necessary. That is also provided by cerebellum. It also has a role of motor coordination that is achieved when the subsequent parts of the same movement or the movement of the several limbs of body parts are combined in a manner that well-timed, smooth and efficient with respect to intended goal. Cerebellum also has a role in motor learning. Here are the loop of motor planning. Motor planning and execution path. This is the external sagittal section showing the position of cerebellum behind the brainstem and fourth ventricle. The brainstem consists of midbrain, pons and medulla oblongata. The dorsal view of cerebellum, it is divided by a primary fissure by anterior and posterior lobes. The ventral view showing the three cerebellar peduncle through which cerebellum is attached with midbrain, pons and medulla. Also showing anterior lobe, posterolateral fissure and flocculonodular lobe. Coronal section of cerebellum showing the deep cerebellar nuclei. From lateral to medial, the nuclei are the dentate nuclei, emboliform and globose nuclei and vestigial nuclei. And it can be remembered by the mnemonic, don't eat greasy food, from lateral to medial. The emboliform and globose nuclei are altogether known as nucleus interpositus. Also showing the core of white matter and grey matter externally, which is three-layered. That is, the cortex is three-layered. The slide showing the functional anatomy of cerebellum. Here the flocculonodular lobe forms the vestibulocerebellum. Nuclei related to it are vestibular nuclei, responsible for balance and movement. Vermis and paravermal area give rise to spinocerebellum. Nuclei related are vestigial nuclei and globose nuclei. There is globose nuclei and emboliform nuclei that are intersposed nuclei. They are related to motor execution. Two lateral hemispheres forms the cerebrocerebellum and related to nucleus in dentate nucleus. They help to control motor planning. It is a three-layered cortex. From below upwards, they are granular layer, Purkinje cell layer, and molecular layer. The cells of granular layer contain granule cell and Golgi cell. Purkinje layer contain big Purkinje cell with their tree-like dendrites projecting towards molecular layer. Molecular layer contain basket cell and stellate cell. Only granule cells of the granular layer releases excitatory neurotransmitter. Other cells are inhibitory in nature. The cells receives input fibers or afferent fibers. Climbing fibers from the inferior olive of medulla and mosey fibers from their brainstem nuclei and the spinal cord nuclei. The afferent or input fibers on their way to cortex gives rise collateral to deep cerebellar nuclei. Mossy fiber stimulate granule cell. Granule cell central process divided and form the parallel fibers at the molecular layer with which the dendrites of Purkinje fiber make synapses. The other cells synapsing each other in a feed-forward mechanism, so that all of the inputs coming into the cerebellum are prohibited from transmitted further. Ultimately, the output fibers emerged from the deep cerebellar nuclei have a message that the included motor action should be precise. Here are the cerebellar inputs. To the vestibulocerebellum or archicerebellum, some form vestibular nuclei and junction of pons and medulla. Spino or paliocerebellum get its input from dorsal and ventral spinocerebellar tract and input from visual, auditory and auditory systems. Cerebrocerebellum or neocerebellum has greatest fiber from the pontocerebellar path, which in turn convey fibers from motor cortex. There are output fibers. Vestibulocerebellum sends fiber to vestibular nucleus. Spinocerebellar output fibers to the globus and emboliform nuclei to the brainstem reticular formation, lateral vestibular nucleus, primary motor cortex, rubrospinal and lateral corticospinal tract. Cerebrocerebellum gives off output fibers to ventrolateral nucleus of thalamus and from there to the cerebral motor cortex. Cerebellar cortex is three-layered. Granular layer contain the granule cells. They are excitatory. Their fibers go to make parallel fibers in molecular layer and synapse there with vertically oriented dendrites of Purkinje cells. Purkinje layer have big cells called the Purkinje cells. This is a picture of Purkinje cell which tremendous amount of arborization of the dendrites. It is one of the most biggest cell of the central nervous system. Molecular layer contain basket cell and stellate cell. All the core of deep cerebellar nuclei are located and acts as outgoing or efferent fibers. Neural circuit of the functional unit. This cell is continually under both excitatory and inhibitory influences. The excitatory influence arises from the direct connection with afferent fibers that enter the cerebellum from the brain or the periphery. The afferent inputs to the cerebellum are mainly two types, one called the climbing fiber type and another called the mossy fiber type. Climbing fibers all originated from the inferior olive of the medulla. After sending branches to several deep nuclear cells, the climbing fibers continues all the way to the outer layer of the cerebellar cortex, where it makes about 300 synapses with the soma and dendrites of each Purkinje cells. This climbing fiber is distinguished by the fact that simple impulses in it will always cause the single prolonged up to 1 second peculiar type of action potential in each Purkinje cell with which it connects, beginning with the strong spike and followed by a trail of weakening secondary spikes. This action potential is called the complex spike. The mosey fibers are all the other fibers entered the cerebellum from the multiple sources, from the higher brain, brainstem and spinal cord. These fibers also send collateral to excite the deep nuclear cell. Then they proceed to granule cell layer of the cortex. These fiber also send collateral to excite the deep nuclear cell. Then they proceed to granule cell of the cortex. There are action divided into two branches that extended 1 to 2 millimeters in each direction parallel to fovea. There are many millions of these parallel nerve fibers. There are some 500 to 1000 granule cells of every one Purkinje cell. It is into the molecular layer that in the dendrites of the Purkinje cell project and 80,000 to 2 lakhs of the parallel fibers synapses with each Purkinje cell. The synaptic connections are weak, so the large number of Mosey fibers must be stimulated simultaneously to excite the Purkinje cells. The activation usually takes place in the form of much weaker short duration Purkinje cell action potential called the simple spike. Here are the complex spike and simple spike pictures. The simple spike is formed by the Mosey fiber and the complex spikes are formed by the climbing fibers. One characteristic of both Purkinje and deep nuclear cell that normally both of them fire continuously. The output activity of both these cells can be modulated upward or downward. Direct stimulation of the deep nuclear cell by both the climbing and Mosey fiber excites them. By contrast, signal arriving from the Purkinje cell inhibit them. Normally, the balance between these two effects is slightly favor of excitation, so that under quiet condition, output from the deep cerebellar nuclei remains relatively constant at a moderate level of continuous stimulation. In execution of rapid motor movement, the initiating signal from the cerebral motor cortex to brainstem at first greatly increases deep nuclear cell excitation. Then another few milliseconds later, feedback inhibitory signal from the Purkinje cell circuit arises. This way, there is first rapid excitatory signal sent by the deep nuclear cell into the motor output pathway to enhance the motor movement. But this is followed within another small fraction of second by an inhibitory signal. This inhibitory signal resembles the delay line negative feedback signal of the type that it is effective in providing damping. That is, when the motor system is excited, a negative feedback signal occurs after a short delay to stop the muscle movement from overshooting its mark. Otherwise, oscillation of the movement would occur. Other inhibitory cells in the cerebellum. In addition to the deep nuclear cell, granule cell, Purkinje cell, two other types of neuron are located in cerebellum. They are basket cell and stellate cells. These are inhibitory cells with short action. Both the basket cell and the stellate cell are located in the molecular layer of the cerebellar cortex. These cells in turn send their action to the right angle across the parallel fiber and causes the lateral inhibition of the adjacent Purkinje cell, thus sharpening the signal. The typical function of the cerebellum is to help provide rapid turn-on signal of the alongside of muscles and simultaneously reciprocal turn-off signal for the antagonist muscle at the onset of the movement. Then the approaching termination of the movement, the cerebellum is mainly responsible for timing and executing the turn-off signal to antagonist and turn-on signal to the agonist. Turn-off signal to the agonist and turn-on signal to the antagonist. This way, the cerebellum do its function through feed-forward and lateral inhibition. What is feed-forward inhibition? Feed-forward inhibition is typically occurs between different brain areas when excitatory neurons excite inhibitory cells, which then inhibit a group of postsynaptic excitatory neurons outside of the initializing excitatory neurons area. What is the difference between feedback system and feed-forward system? Feedback system is reactive, responds to current state of affairs and depends on the input, that is sensory signal. Feed-forward system is anticipatory, depends on the sensory signal but also learns through experience, essential for rapid action. This concludes my lecture. Thank you to all.